And the last talk of this session is by Ure Larsson, who is going to tell us a little bit about the definition and the studies of the immune repertoire.
So I'm measuring more stuff for less money. If you remember a single thing today, the idea is if you give me $25 and just a microliter of serum, I can do essentially the equivalent of 500,000 ELISAs, conceptually speaking, to 500,000 different antigens of interest, with some caveats. So the main idea, of course, is the immune system is a molecular classifier. It's being bombarded with molecules, and you need to make a decision if things are safe or not, self or foreign. The way that it does this, we all know, is through generating a very large repertoire of different receptors. These are non-germline receptors. Everyone has different receptors, including identical twins. Essentially, these receptors can be bound to essentially anything in the universe, in principle. The coolest part about the immune system, I think, or one of the coolest parts, is that it stores a memory of all the things you've seen. So if I could take your blood and generate information from all of the receptors in your blood that's interpretable, I should be able to know a ton of stuff about you. Probably where you're from, different diseases you've had, viruses you've been exposed to, et cetera. You can imagine mining the kind of fossil record of all the stuff you've been exposed to. Even more interesting from our perspective today, we're looking at cancer and precision medicine, is you can imagine that, that you could, by looking at the immune system, decide if someone is going to be a good responder to immunotherapy or not, or whether or not they are going to be at risk for developing immune-related adverse events or something similar to that. So there is quite a large effort going on currently in the community to actually characterize the adaptive immune system in these kinds of contexts by doing things like antibody or TCR sequencing. And in some cases, you actually get some interesting kind of summary information. But the problem is that every individual has different receptors, so it's actually extremely difficult to assign any functional significance to any individual antibodies or TCRs. And because of that, that's one of the main hurdles at actually generating useful biomarkers that are prognostic or diagnostic. And so the assay that we develop in my lab is something that is trying to address this problem directly. And what we actually do is instead of measuring the antibodies and TCRs, we're going to measure the things that the antibodies are binding to. And to do this, we're taking advantage of not only huge advances in DNA sequencing, but also huge advances in DNA synthesis. So let me describe to you how the assay works. Let's say we want to measure if you have antibodies against any possible human protein. So these are autoantigens or maybe cancer antigens. We actually know the sequence of every human protein. And so we actually just download it. We can computationally chop it up into small chunks, like 56 amino acids, 36 amino acids, something like that. We can reverse translate it into a DNA sequence and basically send this text file to a company that a few weeks later sends us back a tube full of DNA oligonucleotides that encode all of these different peptides. So in the case of the human genome, we've done it for it's almost 500,000 different human peptides. We take the oligos, we clone them into a T7 phage display library, and so at the end of this process, which is actually kind of laborious and difficult, what you end up with is a tube that contains a bunch of phage particles, 500,000 different types. Each different phage particle type is expressing on its surface a different human peptide, and inside of that particle is also the piece of DNA that encodes for it. And so now we have a system that you can measure both the protein and the DNA at the same time. The nice thing is you do this one time, and now once you have your phage library, essentially you've done it once, and it's good forever, because the phage can just replicate themselves. So we take our phage library, and I take that microliter of serum that you gave me, I mix them together, and if you have any antibodies that bind to a particular human peptide, then presumably those antibodies will bind to that phage clone. And so then we do an immunoprecipitation, pull down the antibodies, and some of the phage will get pulled down along with the antibodies, specifically the ones that are expressing a protein that you had an antibody specificity for. And then we use next-gen sequencing to sequence all the stuff that's stuck. And so in a single assay, we can basically generate a value for 500,000 different possible peptides. And so we've applied this technology to a bunch of different autoimmune diseases, like rheumatoid arthritis, MS, et cetera. We've discovered a new autoantigen with this. We can see that in RA, people segregate into two different kinds of clusters that's kind of separate from their seronegativity or positivity status, et cetera. But most interesting for today is we actually, we just got this data literally just a couple of days ago, so these may just be random squiggly lines. But we got some serum samples from, for example, Nina's personal vaccine trial, where we know what the individuals are vaccinated against, and we're trying to see if we can track whether they generate an immune response in the antibody repertoire. You can imagine using this to see if people are starting to generate a known autoimmunity against generating diabetes or something like that, looking at the immune repertoire. So actually, we have a human library with 500,000 peptides approximately. We also have a library that contains every possible viral peptide from human infecting viruses and bacterial toxins and strep and staph. And actually, if anybody has an idea for a library that would be useful to them, I would love to work with you and build a new library for you, essentially. So again, $25 approximately per sample, just microliter amounts of serum or breast milk or stool or something like that, and we can generate very, very high-dimensional profiles of the immune response. I'll get to the caveats later if you guys want. So thank you for Mei and Elliot for doing the experiments in the lab, and Tim for analyzing the data. Thanks.
Thank you.